Good day to everyone. I am Bernadette Ramirez and I'm a scientist at the World Health Organization headquarters in Geneva, Switzerland. Kumusta po kayong lahat? I'm very pleased to join you today in this virtual conference. First of all, I'd like to sincerely thank the organizers of this annual conference for inviting me to speak with you today. Like all of you, I am a member of PSBMB myself, and I stand proud as a member of this family. I'm both humbled and honored to be invited to speak here for the Clara Y. Lim Silianco Lecture in honor of one of PSBMB's founders, whose life and significant contribution to the field of biochemistry and molecular biology we will revisit during my presentation. In normal times, I'm sure you would all have wanted to be physically in the same space together. But the COVID-19 pandemic keeps us from doing just that. But there is still so much to be grateful for. I trust that all of you are well and keeping safe wherever you are as you listen in and participate in our convention. At this point, allow me to once more thank the organizers and Secretariat for putting together this year's 47th annual convention. Similar to previous conventions of years past, this event is significant to all of us, as it creates the space for the presentation of current results of our scientific work in our field of endeavor and brings together leading scientists, researchers, and research scholars to exchange and share in a common platform. For this year's Clara Y. Lim Silianca lecture, I have chosen to speak about biochemistry and molecular biology at the interface between health and environment issues. On this slide, you will see the outline of my presentation. For starters, I thought it would be fitting and appropriate to talk about Professor Clara Y. Lim Silianco, her life and her contributions to the field of biochemistry and molecular biology. After all, this special lecture, held year after year at the annual PSBMB convention, was established to honor her legacy. The second and third parts of my presentation will be an overview of the evolution of molecular biology, as it is juxtaposed with the establishment of PSBMB, including some tidbits on our organization. The remaining majority of my talk will use the COVID-19 pandemic as an illustrative example for biochemistry and molecular biology at the interface between health and environment issues. Finally, I'd like to end my presentation with a proposal for a call to action to encourage more engagement of PSBMB and its members in future directions that will ensure that PSBMB will continue to be true to its mandate of research and education and to achieve the bigger goal of utilizing biochemistry and molecular biology for the improvement of the socio-economic and health condition of the country. At the end of my talk, I hope I would have kept you engaged as I walk you through all these presentation items, and I look forward to answering your questions and hearing your comments later. Each year, one of the continuing traditions of the PSBMB convention is the Clara Y. Lim Silianco Lecture, to honor the legacy of our society's founding president. Not all of us had the privilege of personally knowing Professor Silianco, so I thought it fitting to start my talk with a tribute to her, and with the hope that knowing about her life and productive career, we would be given the inspiration, enthusiasm, and perseverance to pursue ours. This slide shows us in a nutshell the life and times of Professor Silianco, spanning all of 9 decades. It all began when she was born in August 1925 in Negros Oriental. Had she still been alive today, she would have been 95 years old. At 20 years of age in 1945, young Clara moved from Gihulngan in the north of Negros Oriental to Dumaguete, which is located in the south of Negros Oriental, to study at Silliman University. At that time, Silliman University was just granted university status in 1938, although it was founded in 1901. Professor Silianco spent 6 years at Silliman University from 1945 to 1951, finishing with a degree in Associate in Arts and a Bachelor of Science degree in Chemistry, magna cum laude. We are privileged to get a glimpse of her personality from an excerpt in the 1949 Silliman University yearbook, which described the young Clara as the honors girl, who takes to photography, reading novels and technical journals, and is unassuming, modest, and talks sense always. Upon finishing her bachelor's degree, she then pursued a master's degree in chemistry at the University of the Philippines, where she also served as a research assistant and instructor in chemistry until her graduation in 1953. After receiving a Fulbright scholarship, she went to the United States to pursue her doctoral studies in biochemistry and organic chemistry at the University of Iowa. While at the University of Iowa, she was also a research assistant at the Department of Pediatrics from 1953 to 1955 and a research fellow in the Department of Biochemistry from 1955 to 1957. Upon obtaining her PhD at the University of Iowa, she returned to a faculty position with the Department of Chemistry and Biochemistry at the UP in 1957. At UP, Professor Silianco pursued a productive career as a prolific researcher, making significant contributions to several fields such as environmental mutagens and anti-mutagens, biochemical, nutrition, and mutagenicity of Philippine medicinal plants. As you'll see, Professor Silianco's lifetime research was also at the intersection of health and environment. Professor Silianco was dedicated to education and wrote textbooks on organic chemistry and molecular biochemistry, as you'll see on this slide. From 1960 to the mid-70s, as an educator, Professor Silianco embodied the popular Chinese proverb which says, "If you're planning for a year, sow rice. If you're planning for a decade, plant trees. If you're planning for a lifetime, educate people." It was in 1973, 47 years ago, when Professor Silianco, together with a group of biochemistry teachers and educators, established then what is known as the Philippine Biochemistry Society, PBS for short, to foster biochemistry education and research in the Philippines. Theirs was a formidable group, led by Professor Silianco as the founding president. PBS was committed to upgrade biochemistry in the country by encouraging research and education among its members as well as biochemists outside of the country. The PBS mission at that time, as it stands true today, is to utilize biochemistry for the improvement of the socioeconomic and health conditions of the country. In 1994, at the age of 69 years old, Professor Silianco was bestowed the rank and title of National Scientist of the Philippines, or what is called as the Pambansang Alagad ng Agham ng Pilipinas. It is the highest award accorded to Filipino scientists by the Philippine government, and this is in recognition of her distinguished achievement in science and technology. Professor Silianco died at the age of 88 years old in July 23, 2013. The words of Silliman University President Dr. Malayang sums it all. Professor Silianco was the face of a continuing pursuit of broader horizons for science and its potential in bringing sustainable development for the Filipino nation. Professor Silianco may no longer be with us. However, as the saying goes, the goal is not to live forever anyway. The goal is to create something that will. Let me leave this slide with this thought. A teacher affects eternity. Thus, a teacher can never tell where his or her influence stops. The song may have ended, but the melody lingers on. We now move on to another timeline, the history of molecular biology, spanning decades from its early beginnings in the late 1920s. This slide shows some of the significant milestones, which I won't go into for specific details. The broad headings are spelled out on this slide: the discovery of DNA, genes and DNA, DNA, RNA, and protein interactions, DNA cloning and genetic engineering, and the genome era. For the interested audience, additional details can be found in relevant textbooks and other references. The reason I put up this slide is to juxtapose the establishment of PSB in 1973, which was later renamed 20 years later, in 1993, as the Philippine Society for Biochemistry and Molecular Biology, with the evolution of molecular biology as you see at the bottom of this slide. Why this is important is because I'd like to highlight the achievements in molecular biology during the era of genetic engineering and the genome era, side by side with the evolution of PSBMB. I consider myself extremely fortunate to witness within my lifetime the great strides and technological advances in molecular biology. More importantly, I think as chemists, biochemists, and molecular biologists, we should consider ourselves privileged to be given the opportunity to participate in shaping a future that we want to live in and to live for, as we participate in the further evolution of our field and as we responsibly apply the new technologies. Therefore, the take-home message on this slide is for us to reflect on what I consider as the biggest challenge faced by members of PSBMB and to be in our field. Our biggest challenge is neither that of teaching about the history of our field nor the lessons learned from history. It is about why history matters. I believe history is there to ignite that first spark in each and every one of us, and that which should propel us across the zigzagging and interconnecting linkages of the past, the present, and the future. When we are without the knowledge of the past history, we are like trees without roots. This slide is about PSBMB and the passage of time. On August 1973, the Philippine Biochemistry Society was established by a group of biochemistry educators to foster the development of biochemistry in the Philippines. In 1993, the general membership of PBS voted to change its name to what we know now as PSBMB during the annual convention held that year. This name change underscores the integration of molecular biology as a closely related discipline with biochemistry. Also in 1973 onwards, PBS is a member of FAOBMB and a representative of IUBMB. From 1978 to 1993, PBS published the Bulletin of the Philippine Biochemical Society. In 1995, the Young Scientists Forum was established and has since been a platform for showcasing undergraduate student research. The year 2004 saw the establishment of the first Review and Update Workshops series, what we call RevUp for short. It is for continuing education and to improve and update the teaching of biosciences in the primary, secondary, and tertiary levels. The first RevUp was held at Silliman University in Dumaguete in 2004. Yearly RevUp workshops have been held since it first started and have been brought to various locations in the Philippines. In Manila, Angeles City and Pampanga, Legazpi and Albay, Iloilo, Bukidnon, Nueva Ecija, Bacolod, Ilocos Norte, Quezon, Cebu, and General Santos City. In addition to RevUp, PSBMB also conducts what is called as REDUP, which is the acronym for Review in Education and Updates in Biochemistry and Molecular Biology. The first REDUP was held in 2018. Recently, in 2016, PSBMB signed a Memorandum of Agreement for collaboration with the Institute of Molecular Biology Academia, Sinica, in Taiwan. It will be interesting to get an update on the status of this collaboration from the PSBMB Board of Directors. In 2017, the first PSBMB Central Luzon Chapter Symposium was held. As part of its regional outreach, I understand that PSBMB now has several active regional chapters based on geographic clusters. In Northern Luzon, in Central Luzon, Southern Luzon, Visayas, and Mindanao. Each chapter has its own set of officers, a president, a vice president, a secretary, and a treasurer. And the chapter president sits as a member of the PSBMB Board of Directors. For the current year 2020, PSB convention goes for the very first time as a virtual convention and is hosted by the local organizers from the PSBMB Central Luzon chapter led by its president, Dr. Casiano Choresca. As I have earlier mentioned, PSBMB throughout its 47 years of existence is committed to its mandate. And we'd like for us as members to see that we are also committed to this mandate of establishing communications and exchange of professional knowledge among its members and other biochemists abroad. We now move on to the next segment of my presentation. This is about the COVID-19 pandemic, which we will use as an illustrative case for biochemistry and molecular biology, being at the interface between health and environment issues. Let's start off with this food for thought from a researcher and specialist in the replication of RNA viruses such as coronaviruses. He said, "Our relationship with nature plays a key role in the development of this type of pathogen, the new coronavirus. Nature has been seen as a hive that one could pilfer without restraint. Global anthropogenic pressure has favored the emergence of viruses that were until now hidden in animals and maintained in their natural habitats by high levels of biodiversity. The coronaviruses that cause SARS, MERS, and now SARS-CoV-2 all came from the animal world and crossed the interspecies barrier. At the beginning of the COVID-19 pandemic, we were confronted with an enemy that we cannot see and that we do not know much about. Scientists started asking the question, how do we fight such an enemy? The only way is to study and learn more about it, what it wants, what it does in order to survive, and where its weaknesses lie. And this is where science played a significant role. This COVID-19 pandemic has brought to our attention how building up scientific temperament and propagation of scientific culture are critically important to tackling a pandemic of this scale. On this slide, I would like to bring you back in time during the latter part of 2019, at a time when we had no inkling on how the current pandemic was going to change our lives and the rest of humanity. As you can see on this slide, it was most likely in October-November last year when there was evidence to suggest that what we now call the SARS-CoV-2 virus, and which may have descended from a coronavirus that infects wild bats and have spread to humans through an intermediary wildlife host. As you see here, a link with the environment. Very clear that molecular biologists were also at the forefront of undertaking phylogenetic studies to understand the virus. On the 1st of December that year was the earliest date for which symptom onset was reported. This may well be our patient 0. Later, there was a report of a patient with pneumonia-like symptoms, and finally on December 16, we heard of the first documented COVID-19 hospital admission. Once more, on the environment side, sewage samples collected from wastewater treatment plants in Milan and Turin in Italy showed the presence of SARS-CoV-2. By 23 December, a bronchoalveolar lavage sample from Wuhan, the epicenter, was sent to Vision Medicals in Guangzhou for metagenomic massive parallel sequencing analysis. So as you'll see, science was front and center from the early beginnings of this pandemic and contributing to the understanding of the virus from the environment as well as from the human patients affected by it. This slide is a bit busy and I won't go through each of the individual bullets. I have conveniently highlighted some points to be stressed here. First, by January 12, China had already publicly shared the genetic sequence of the new virus. This early release is very important and was instrumental for R&D processes to take place immediately for the development of diagnostic test kits with high performance accuracy. By the 30th of January, WHO had declared the novel coronavirus outbreak a public health emergency of international concern. This was followed by WHO action to convene the Global Research and Innovation Forum on February 11 and 12. At this same time, the R&D blueprint was activated and the disease got its name, COVID-19. With the activation of WHO's R&D blueprint, coordinated efforts were initiated across the globe to understand COVID-19 and to prepare countries to respond. I will talk more about the R&D blueprint in a bit in the next slide. By 11 March, WHO had assessed the situation as a global pandemic. 2 days later, on March 13, the COVID Solidarity Response Fund was set up for donations from private individuals, corporations, and institutions to support the global response to the pandemic. And the rest of the events that followed, as we all have witnessed, went so quickly, resulting in lockdowns in almost all countries affected by the pandemic. On this slide, I'll give you a brief on what the R&D blueprint is about. For those of you who may be interested in learning more, you can follow the link I provided at the bottom of this slide. As I have mentioned in the previous slide, the R&D blueprint was activated on February 11 and 12 during the Global Research Innovation Forum. This activation signaled a call to scientists to step up to the plate, to accelerate diagnostics, therapeutics, and vaccines for the new coronavirus. Needless to say, this is an unprecedented program. To top it all, this was also a time for strengthening channels for information sharing between countries. The aims were to improve coordination between scientists and global health professionals, accelerate the research and development process, and to develop new norms and standards to learn from and improve the global response. Urgency became the name of the game, and scientists had to close ranks, combining scientific knowledge from previous infections with modern scientific and technological advances. On this slide, we will talk about diagnostics. Diagnosis is made using a combination of clinical, laboratory, and radiologic features. By now, the rest of the world are all too familiar with the term RT-PCR, a term that had been previously reserved for use in the lab by molecular biologists. The figure on the right-hand side of this slide shows the critical roles of the clinical laboratory in managing COVID-19, from molecular testing, serology, and biochemical monitoring. I have provided a link so that anyone interested in the audience can get more information on the key considerations for laboratories. I've also provided information to the latest up-to-date publication reviews on laboratory techniques on diagnosis of COVID-19. As you may be aware, newer tests have now been developed for laboratory diagnosis. The latest test I'd like to draw your attention to as an example is the Lucira COVID-19 all-in-one test kit, a molecular real-time loop-mediated amplification reaction single-use test with results in just 30 minutes or less. This new test is authorized by the US FDA under the administration's emergency use authorization. It is intended for prescription home use with self-collected nasal swab samples. For individuals aged 14 years and older who may be suspected of COVID-19 by their healthcare provider. As I have mentioned in the previous slide, additional test systems other than the conventional RT-PCR continue to be developed. This slide mentions a few of those, from using CRISPR to isothermal nucleic acid amplification technologies to molecular microarray assays to serology testing. These are all summarized in recent publication reviews as shown on this slide. If you're interested in learning more about the target product profiles for priority diagnostics to support response to the COVID-19 pandemic, WHO also has a publication for that, and I have provided a link to it on this slide. Let's now move on to talk about therapeutic strategies. The approach was to understand how the virus replicates inside the host cell and to develop therapeutic agents that could block this replication cycle. Because of the urgency of the situation, scientists pursued the investigation of drugs, which are already approved and available commercially and can be repurposed to target the new coronavirus. On March 18 this year, WHO and its global partners launched the Solidarity Trial, which I'm sure you already have heard of. It is an international clinical trial that aims to generate robust data from all over the world in order to find the most effective treatments for COVID-19. The research design was through a randomized clinical trial, which we all know normally take years to design and to implement. But the Solidarity Trial was specifically established to accelerate this process, which helped facilitate the rapid, worldwide comparison of unproven treatments and to generate the strong evidence needed to determine the relative effectiveness of potential treatments. Results are now available for the WHO Solidarity Therapeutics Trial. Please see the links that I have added onto this slide for more details. The interim results were from 30 countries, 10,000 patients in 500 hospitals. The results indicated that remdesivir, hydroxychloroquine, lopinavir, ritonavir, and interferon regimens appeared to have little or no effect on 28-day mortality or during the in-hospital course. What is significant, though, about the Solidarity Trial is the demonstration that large international trials are possible, even during a pandemic. This approach offers the promise of quickly and reliably answering critical public health questions on therapeutics. Moving further on, let us now talk about vaccines against COVID-19. Vaccines typically require years of research and testing before reaching the clinic. But for the current pandemic, we have seen how scientists are racing to produce a safe and effective coronavirus vaccine. This speed is unprecedented in vaccine history. As of 23 November, researchers are testing 236 vaccines in development, with 40 vaccines in clinical testing on humans, and at least 87 preclinical vaccines that are under active investigations in animals. As you can see on this slide, there are at least 10 leading vaccine candidates in the pipeline. If you are interested in the target product profiles for the COVID-19 vaccines, please click on the link I have provided on this slide. This document details the preferred and minimally acceptable profiles for human vaccines for long-term protection of persons at risk of COVID infection. On this slide, you'll see the different types of vaccines under development. There are basically 6 types of COVID vaccines. We have the live attenuated virus type, inactivated virus, protein subunit vaccines, virus-like particles, DNA and RNA vaccines, and viral vector vaccines. Of those undergoing clinical trials, the most common type is the protein subunit, followed by the viral vector, then followed by DNA, RNA, inactivated virus, virus-like particle, and live attenuated virus. This slide shows us the early front runners in the race for a COVID vaccine. In recent weeks, we have heard promising news from the phase 3 clinical trials of 3 vaccine candidates. These are namely from Pfizer-BioNTech, Moderna, and the Oxford-AstraZeneca vaccine. Both the Pfizer and Moderna vaccines are mRNA vaccines, while the one from Oxford-AstraZeneca is a viral vector vaccine. Preliminary results are encouraging as far as protection is concerned. These levels are way above the preferred or critical or minimal efficacy required in the target product profile, which is at least 70% for preferred efficacy and around 50% for minimal efficacy. On this slide, you will see a comparison of the 3 vaccine front runners as far as product stability and storage is concerned. This is an important consideration since this has implications for use of distribution and availability of the vaccine product. If you are wondering how access for diagnostics, therapeutics, and eventually vaccines is coordinated during the pandemic, the answer is on this slide, the ACT Accelerator. On this slide and the next 2 slides after this, I'll be talking about the ACT Accelerator program, which is also the short name for Access to COVID-19 Tools. ACT was launched in April 2020 by the WHO, the European Commission, as well as France. As the name implies, ACT Accelerator was set up and brings together governments, global health organizations, manufacturers, scientists, private sector, civil society, and philanthropy to contain the COVID-19 pandemic faster and more efficiently by coordinating the equitable access of diagnostics, treatments, and vaccines across all countries. Let's now focus on vaccines. At an early stage during this pandemic, it quickly became apparent that to end this global crisis, we don't just need COVID-19 vaccines. We will also need to ensure that everyone in the world has access to them. This has triggered the global leaders to call for a global solution. And the solution is called COVAX, which is led by 3 institutions: the World Health Organization, the Global Vaccine Alliance, or Gavi, and the Coalition for Epidemic Preparedness Innovations, CEPI. In the event that a safe and effective vaccine is found and authorized, COVAX, the vaccine pillar of the ACT Accelerator, will facilitate the equitable access and distribution of the vaccines in all countries, in all corners of the world, regardless of their wealth. COVAX will achieve this by acting as a platform that will support the research, development, and manufacturing of a wide range of COVID-19 vaccine candidates and negotiate their pricing. All participating countries, regardless of income level, will have equal access to these vaccines once they are developed. The initial aim is to have 2 billion doses available by the end of 2021, which should be enough to protect high-risk and vulnerable people, as well as frontline healthcare workers. These are the priority recipients of the vaccine. Therefore, the COVAX is literally the lifeline and the only viable way in which lower income funded nations who would otherwise be unable to afford these vaccines will get access to COVID-19 vaccines. The same goes for higher income self-financing countries that have no bilateral deals with manufacturer. COVAX is going to be the solution. The COVAX facility is the global procurement mechanism of the COVAX. I will not go into details of how this procurement mechanism will be implemented, but suffice to say that the COVAX facility will be guided by a fair allocation framework. Please see the link provided to be able to read more about the COVAX and the COVAX facility. The COVAX Advanced Market Commitment is the financing instrument that will support the participation of 92 LMICs and LICs. These are the low and middle income countries and the low income economies that are in and within the COVAX facility. At the moment, the AMC requires an urgent investment of $2 billion from donors. We now leave the topic of vaccines and move on and back to talking about the environment through this slide. The importance of commercially raised animals in the COVID pandemic has received much attention, particularly during the past few weeks. It turns out that minks are among the animals that can be infected with coronaviruses. There had been a report of a new variant of the coronavirus that was detected in farmed mink. For those of you who may not be familiar with mink animals, these are dark-colored, semi-aquatic, carnivorous mammals. They are highly prized for their fur for the use in the manufacture of warm clothing. Because of high demand for mink, hunting has given way to farming. The mink link is of major concern since minks can serve as an alternative host to promote mutations of the SARS-CoV-2, which can be passed on to humans and other animals. I believe this is my final slide. From my presentation today, we have seen science in action and at the forefront in the response against COVID-19 pandemic. The role of science is not only true against COVID-19, but for other public health and environment issues, which I'm sure everyone here in this room are addressing. Science is clearly front and center. Science can help save lives, and science gives us hope for the future. At the end of my presentation, I'd like us to rethink our roles and how we can contribute to society and sustainable development. I'd like to propose a call to action for us at PSBMB. For us to undertake an assessment of our roster that includes both local and international network of scientists that will lead to a stronger community of practice for collaboration, mentorship, and capacity building. We can now dream of PSBMB Global instead of just PSBMB Local. We'd like to also encourage ourselves to go for an outreach campaign that reaches out to various sectors and the public. For example, we can initiate an information drive to promote vaccines and to counter vaccine denial, or an information drive responding to infodemic and misinformation. And finally, another call to action for an advocacy strategy for a specific policy issue. For example, funding and infrastructure support for research. With this, I'd like to end my presentation. Many thanks for your kind attention. Maraming salamat po.